In this video, we'll discuss the second phase of photosynthesis, the Calvin cycle. All right, before the Calvin cycle can take place, we have to have stored energy in the form of ATP and NADPH. Recall that we stored that energy during the first stage of photosynthesis, the light-dependent reactions. Now the chloroplast will use that energy to synthesize a carbohydrate. First, let's make a point of recalling that the carbon to make organic molecules comes from inorganic carbon dioxide. The chloroplast will fix this carbon dioxide, and when we say fix, we mean that it will incorporate the inorganic carbon dioxide into an organic molecule. So plants and other photosynthetic organisms are taking carbon dioxide out of the atmosphere or the water in which they live and incorporating it into the living organisms of the biosphere. We can divide the Calvin cycle into 3 parts, and we won't cover all the reactions of the Calvin cycle. We'll pretty much just describe the major phases and the important parts of each. All right, so first of all, this is a cycle where it starts and ends with the same molecule. This is really similar to the citric acid cycle where we start and end with oxaloacetate. Here, however, we're going to start and end with a 5-carbon molecule called ribulose 1,5-bisphosphate, and we abbreviate that as RuBP. Our 3 major phases of the carbon cycle are carbon fixation, reduction, and regeneration of RuBP. And you'll notice that there are numbers of molecules specified here. We're using the number of molecules that will be necessary to produce 1 molecule of glyceraldehyde 3-phosphate that can be used to make glucose and other carbohydrates. And just to jump to the punchline, we'll have to produce a total of 6 glyceraldehyde 3-phosphate molecules to get 1 that we can actually use. More on that in just a bit. We'll start with the first stage of the carbon cycle, which is carbon fixation. This is accomplished by a really important enzyme. Its nickname is Rubisco. This stands for ribulose 1,5-bisphosphate carboxylase oxygenase. And even though that name is really long, the enzyme does what its name suggests. It takes carbon dioxide and adds it to a molecule that we've already met, ribulose 1,5-bisphosphate, or RuBP for short. Rubisco is probably the most abundant enzyme on Earth, and we could also argue that it's the most important, because without it, the most essential step of photosynthesis could not take place. This reaction produces a 6-carbon molecule because RuBP has 5 carbons and carbon dioxide has 1, but the 6-carbon molecule is immediately broken into 2 3-carbon molecules called 3-phosphoglycerate. Now we have the second stage of the Calvin cycle, which is reduction. 3-phosphoglycerate is reduced, meaning electrons are added. And these come from the molecules which were produced in the light-dependent reactions, ATP and NADPH. Once reduction is done, we're left with glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate is the major product of the Calvin cycle, and it can be used to make glucose. It takes 2 glyceraldehyde 3-phosphate molecules with 3 carbons each to make 1 molecule of glucose, which has 6 carbons. And for every 6 total G3P molecules that the cell makes, 5 of them have to be used to regenerate the starting molecule RuBP. So we can only use 1 out of every 6 produced to make glucose and other organic molecules. The rest have to proceed through the rest of the Calvin cycle and stage 3, which is regeneration of RuBP. And if we think about the math and the carbons, this makes sense, right? We take 5 3-carbon molecules, because remember we've subtracted 1 to go make glucose, and we take these 5 3-carbon molecules, glyceraldehyde 3-phosphates, and rearrange them to make 3 5-carbon molecules, which is ribulose 1,5-bisphosphate, or RuBP. All right, but this cycle right here, the Calvin cycle, this is the series of reactions that's responsible for just about all of the biomass of plants, algae, bacteria, archaea, animals, fungi, and protists, almost all of the biomass in the whole biosphere.